All right, so in this lecture of the genetics unit, we're going to go over some high-yield examples of aneuploidies. So first, an overview. What is aneuploidy? It's when the number of chromosomes differ from the normal number of 46. So if you remember, you get 23 chromosomes from your mother. So we'll say this is the egg, and then you have a sperm that comes in from your dad that has 23. And then fertilization occurs, and you have a zygote with 46 total chromosomes, 23 pairs of chromosomes. Here's your 23 pairs here, and so this is normal. You have 46 total chromosomes. Now, monosomy is when you have a loss of a chromosome. Could be one chromosome, could be more than one chromosome. So in this case, we've lost one chromosome, so now we have 45 total chromosomes. And then trisomy is the gain of a chromosome, and so in this case, you can see we've gained a chromosome, so now we have 47 chromosomes. And as a result of this, when you lose or gain a chromosome, that's when you can cause these genetic syndromes that we'll talk about in the next few slides for the rest of this lecture. And the last thing I'll point out is that this last pair, this 23rd pair, this is your X and Y chromosome if it's a male, or it's your XX if it's a female. So this is the sex chromosomes, is the last, or the 23rd pair. So the first disease we'll go over is trisomy 13, or Patau syndrome. Now for each of these diseases, we show you this convention here of how to write out the chromosome corresponding to the disorder. So 47 is the total number of chromosomes. This is the sex chromosomes, so whether it's a XY or XX combination, and then this plus with this number corresponds to where you have an extra chromosome. So here you have an extra 13th chromosome, so plus before the 13. So 47 total chromosomes, XY if it's a male, extra chromosome in the 13. Here 47 female, extra 13th chromosome. So this is a rare disorder, even among aneuploidies, and it may be due to meiotic non-disjunction or translocation. So you remember meiotic non-disjunction. So during meiosis, you have chromosomes here that are lining up during metaphase, and then you remember during anaphase, you have that mitotic spindle that pulls them apart because you want to evenly distribute them into the cells when they divide, so this way and this way. The problem is if non-disjunction happens, there's no separation at the centromere, and so one side gets both chromosomes and the other cell gets none, and so then this ends up splitting into where essentially you have one chromosome, this guy here, then you have two there, and then you have one there, and then you have a cell like this where you have one here, and then you have 0 here. And so if this cell combines with one that only has this, then you're going to end up with, and you can see how this happens. So you have normal pair here, and then you have an extra one here. And in this case, it would be 13. Translocation, if you remember, is where you're basically just exchanging pieces of chromosome to another chromosome. So if you have two chromosomes like this part here, this portion could exchange with that, and so you're shifting all the genetic material with that. And so what ends up happening is you have extra pieces of chromosome 13 in other areas. At the end of the day, regardless of which cause it is, this extra genetic material can cause devastating and complex defects across numerous organ systems, which we'll talk about here in a few slides. So the labs, this can actually be pretty high yield on exams. So during the first trimester, so you got to pay attention to what trimester it is, of pregnancy, you'll have decreased human chorionic gonadotropin, or HCG. So the big ones are HCG, and decreased pregnancy-associated plasma protein, or PAPPA. So both of these will be decreased during the first trimester. Clinical features, so they'll have retardation, holopresencephaly. So if you remember, this is failure of the forebrain to develop into two hemispheres. Remember, the brain is essentially two hemispheres like this for the brain, so it doesn't divide and form these two. Just for review, the forebrain during neurodevelopment, it divides into the diencephalon and the telencephalon. And the telencephalon forms the cerebrum, which then forms the cerebral cortex, and the diencephalon forms couple structures, namely the thalamus and the hypothalamus. And this can vary in terms of severity. Sometimes it's really horrible, and then other times it's not as bad. Often, if it's really bad, it's not often even compatible with life. So these patients, they can also have cleft lip, cleft palate, polydactyly, which is extra fingers, small eyes or abnormally shaped eyes, congenital heart disease, mainly ventricular septal defects, and patent ductus arteriosus or PDA. They can have renal defects, cutis aplasia. This is where you have missing skin or hair in a certain portion on the body, rocker bottom feet, and then often this is fatal in the first year of life. All right, so trisomy 18, also known as Edwards syndrome. So the convention here is, again, 47 total chromosomes because you have an extra 18. This is how it'd be written for an XY combination for males, XX for females. This is also rare, but it's more common than Patau, but less common than Down syndrome. So as far as frequency in the population, Downs is the most common, then Edwards, and then lastly, Patau. And one way you can remember that is that 21 is larger than 18, which is larger than 13. Just like trisomy 13, this may be due to meiotic non-disjunction or translocation. So we explained that for the previous one. Same mechanism here. The labs, so the labs are a little bit different here. So this one you got to look into the second trimester. So you got the first here. So similar to trisomy 13, decreased HCG, decreased PAP-A. So this is the same as 13. However, the difference is for trisomy 18 is in the second trimester, you do what's called a quad screen. So that would be HCG, alpha-fetoprotein, estriol, and then inhibin A. So that's the 4 components of a quad screen. So in the second trimester, you're going to have decreased HCG, decreased AFP, decreased estriol, and then decreased inhibin A. So as far as the first trimester goes, it's the same as trisomy 13. Get into the second trimester, this is where you differentiate it. Clinical features, so they're going to have retardation as well. So again, having this extra genetic material causes devastating effects on the development of multiple organ systems. Enlarged occiput, so this is the occipital bone, this posterior bone in the skull. So you could have this enlarged. Sometimes that's called an occipital bun. Migragnathae, this is a small jaw, so underdeveloped jaw, and then lower ears. Characteristic of this disease is overlapping fingers, often the second or third digits. So you can see that in the picture here. Here's the second digit here overlapping with the third, fifth overlapping with the fourth. Also clenched fists, and you can see that here with this infant's fist that's clenched. Congenital heart disease, often ASD, atrial septal defect, ventricular septal defect, and then PDA. Rocker bottom feet, and then like trisomy 13, this is often fatal within the first year of life. So trisomy 21, also known as Down syndrome. So they have 47 chromosomes because they have an extra 21st chromosome. This is the most common genetic cause of intellectual disability. 1 in 700 incidents. It's the most common trisomy, which we've discussed previously here. And it may be due to essentially one of 3 mechanisms: meiotic non-disjunction, as we've talked about, translocation, or mosaicism. So mosaicism, real quick. So during embryonic development, you have cells that are dividing, right, to form the embryo as it grows. And so you keep dividing and dividing. And so what could happen is that mosaicism is essentially 2 different cell populations or 2 different cell lines within one individual. And so you have cells dividing here. So this guy keeps dividing normally, but let's say there was a mutation that occurred here, or a non-disjunction, which this would be mitotic non-disjunction. So in this case, we had mitotic non-disjunction, because that can happen during mitosis. And so then you have 2 populations of cells, and these guys keep dividing and dividing. Meanwhile, these normal cells in blue here also keep dividing. And so when it's all said and done, you have an embryo that's composed of 2 different types of cells. You have the normal cells and then the cells affected by non-disjunction or some kind of mutation. And the breakdown really is the vast majority, about 95% or so, is due to this. About 4% maybe is due to this, and then 1% is due to mosaicism. So the labs, first trimester of pregnancy. So this is unique. You have increased ACG, decreased PPA. So if we do our little table here, we have the first trimester, second, so increased HCG, decreased PAP-A, and so that differentiates it from trisomy 13 and trisomy 18, because in those, both of these are decreased. Ultrasound will show nuchal translucency and hypoplastic nasal bone. Sometimes you'll see this in a question stem. They're tipping you off to Down syndrome. Second trimester of pregnancy, so you have that quad screen again. So in this case, they're also going to have increased HCG, decreased AFP, which is similar to trisomy 18, decreased estriol. However, they're going to have increased inhibin A. So this is unique, and this is unique. Here's a summary table. This isn't in your book, but you may want to draw this out into your book or copy this down. Just gives you a good comparison. We have each syndrome here, the chromosome affected, and then by trimester here. And so you remember, big thing here is Down syndrome versus Edwards. That ACG is elevated. Also that inhibin A is elevated. Downs versus Edwards and Patau in the first trimester, increased ACG. These ones, you got to go to Edwards and get that quad screen, and they'll all be down. And so that's how you can distinguish Edwards from Patau. Clinical features of Downs, there's a lot of them, and often many of these can show up on exams. So like we said, intellectual disability, hypotonia. They have some unique facial features. Here's a picture of a baby with Down syndrome. So they have a short, flat face and nasal bridge. So you can see that here, a single palmar crease. So this horizontal single crease here like this, as you can see. Enlarged epicanthal folds. So here's a picture of that here. And this is essentially a extended fold off of the upper eyelid that covers the corner of the eye. And you can see that here. You can sort of see it here as well, where the patient also has it as well. And then upslanting palpebral fissures. So that's essentially where their eyes are upslanting like this. You can see they're kind of tilted in like this. And then they have upslanting like that. So if you drew a line like that, it would be up like that. So that's what that means. So these are all these facial features. So some more clinical features, duodenal atresia. So if you remember, the duodenum is the first part of the small intestine. It comes off the stomach like this, wraps around the head of the pancreas like this. And then so you have atresia where it has a dead ending essentially. And so as you can imagine, that can cause significant problems. Congenital heart disease, they can very often have ASD, VSD, atrioventricular canal defects, endocardial cushion defects as well can occur in people with Downs. Brush fields, so gray, white spots on the iris. This is also incredibly high yield for Down syndrome. They have increased risk of ALL, so acute lymphoblastic leukemia, and then increased risk of acute myeloid leukemia. These can often be questions on tests. This is also interesting and also very high yield, increased risk of early onset of Alzheimer's because amyloid precursor protein, APP, is on chromosome 21 as well. So if you have an extra 21st chromosome, you're going to have increased levels of this protein, which then build up and lead to Alzheimer's. And then they'll also have a protruding furrowed tongue, which essentially means they have a number of these horizontal lines or indentations. That's probably the word, indentations of the tongue like this. All right, so briefly we'll walk through what a Robertsonian translocation is. This can be a cause of Down syndrome. So a Robertsonian translocation is a specific type of translocation when two short arms of chromosomes are lost and the two long arms fuse at the centromere. So in a lot of chromosomes, both arms are about equal length. And when we talk about two arms, it's the two ends joined at the centromere like this. However, there are chromosomes, specifically these ones, 13, 14, 15, 21, and 22, which is why they're most likely to be involved in a Robertsonian translocation, is they have a short arm and a longer arm. So these are what they're called achocentric chromosomes because one arm is much shorter than the other arm. Another thing to note is that the information contained in the shorter arm is often repeated information. It's not as important or not very important at all because it's repeated elsewhere in the genome in other chromosomes. And so if it's lost, it's not a big deal. So what happens here is you have this will fuse with this and then this will fuse with that. And so you get these two short arms fused together and then you get a long arm and a long arm fused together. Now, this usually disappears during the process of meiosis, and that's not a big deal again because this information is repeated. Now, when this cell divides, there's a number of unique combinations that can be found because, remember, you would have duplicated these and these. So you have an extra one here that forms, and then you have an extra one here. And then instead of having another normal 14 or a second normal 21 to divide, because you normally would have this, right? And you would divide 1 into 1, and 1 into 1 like this, right? And so instead, because of this translocation, you only have one copy of the 14, one copy of the 21, and then you have this fused chromosome that's a result of the translocation, fused long arms, and then you have fused short arms here. And so this is what's going to be divided. Now, there's a number of combinations that we'll go over in the next few slides. So the first result is you could get where you have normal. So you have a 14 and you have a 21, and then you have the fused long arms, and then you have the fused short arms. So that's one combination that could result from this. Another combination is you could have the fused long arms combined with the 14, and then you have the normal 21 with the short arms. The last one is you have the normal 14 with the short arms, and then you have the long arms with the normal 21. And now we'll go through each of these and show you what the final result will be when any of these are fertilized by a normal gamete. So the first combination here, you have a 14 and you have a 21, so this is normal because you have 2 copies of 14, 2 copies of 21. This is a totally normal individual. Now for this cell, you have this fused long arm, fused short arms. This is going to get lost in the process of meiosis, so it will get fertilized by a normal gamete that's carrying a normal copy of a 14 and a normal copy of a 21, and so this comes down and becomes a part of that, and so this is what you have. However, this is a balanced carrier because, remember, the long arm is where the important genetic material is for both 14 and 21, so you essentially still have 2 copies of 14 and 2 copies of 21. And so this is what's called a balanced carrier, and this is actually a normal phenotype. Because again, these short arms carry repeated genetic material. Repeated meaning the material is also contained in other areas of the genome as well. Now the second combination. So you have this normal 14 with the fused long arms, fertilized with a gamete that has a normal 14 and a normal 21. So you'll have 2 copies of a normal 14 chromosome, and then you have 1 copy of a 21. Now what happens is you actually have 3 copies of 14 because this long arm is essentially the same as a full chromosome because this short arm isn't as important because it contains that repeated information. So you essentially have 1, 2, 3 copies of 14, hence trisomy 14, and then you have 2 copies of 21 because you still have this long arm of 21. So this on this side it's normal, but on this side you have 3 copies of 14. Over here you have these short arms that are fused. This is going to disappear. You're going to be fertilized with a normal gamete, and so you'll have 2 copies of 21 and only 1 copy of 14, so monosomy 14. Last combination here, so you have 1 14 here, and then you have the short arms here. This disappears in meiosis. Fertilized by a normal gamete, you get 2 copies of 14, 1, 2, and then you only have 1 copy of 21. Over here you have the fused long arms and then a normal 21. So you're going to fuse with a normal gamete, and this is how you get trisomy 21. Because again, you have all the important genetic information from 21 contained in here. This information isn't as important. So you essentially have 3 copies of chromosome 21, and then here you have the important information for 14, so you have 1, 2 copies of 14. And so again, these 3 copies of the important information from 21 gives you trisomy 21. So just to recap here, it can be balanced or unbalanced. So a balanced translocation is a normal phenotype usually. So this is balanced because you have 2 copies of 14 and 2 copies of 21. Here you have a fused long arm chromosome here, but since you have the important information from 14 pairing with a normal 14 chromosome, and then you have the important information on the long arm for 21 pairing with a normal 21 chromosome, you still have 2 copies of the 14 chromosome and essentially 2 copies of the 21 chromosome. So this is a balanced carrier and this is normal. Now, however, This is what's called an unbalanced translocation, and this will cause problems such as Down syndrome. And you have a normal chromosome 14 paired with a long arm from a 14, so you have the normal amount of 14. However, you have this long arm of chromosome 21 paired with 2 copies of chromosome 21, so you have 3 copies, gives you trisomy 21, and that gives you Downs. So now to round out this lecture, we're going to talk about some examples of aneuploidies that affect the sex chromosomes, so X and Y. So just the convention, so in Klinefelter, you have an extra X chromosome. So that's why you show this here. There's no plus here because there's no extra 17th or 21st chromosome, and so there's no point in putting that. You just have an extra X chromosome. This is often due to meiotic non-disjunction. Now remember with the sex chromosomes, in males, you have XY, and then it gets split up into X and Y. And then in females, you have 2 X's, and then it gets split up into 2 X's. Now, what can happen is if it's due to paternal non-disjunction, is you have an X and a Y like this. And so what happens is you have XY like this, and then nothing here, and so then this combines with a normal X from a female. Fertilization occurs, and you have XXY here. On the maternal side of things, you have non-disjunction in the mother. You get 2 X's here and nothing here, and so this combines with a sperm with a Y, and so that gives you, again, XXY. So first let's review some normal male endocrine physiology here. So you have the hypothalamus here. Which releases gonadotropin releasing hormone, which stimulates the pituitary to release 2 different hormones. FSH, which then goes on and stimulates their Sertoli cells. And then it also stimulates spermatogenesis. And then the Sertoli cells release a substance known as inhibin. Now, inhibin comes back and negative feedbacks on the pituitary, inhibiting release of FSH, and it also has negative feedback on the hypothalamus, inhibiting release of gonadotropin releasing hormone. So it's a classic example of feedback inhibition. Now, the other hormone produced by the pituitary is luteinizing hormone, which then goes on and stimulates the Leydig cells to produce testosterone. Now, testosterone is responsible for normal male sexual development and other male characteristics throughout the body. But in this case, what we're concerned is that testosterone comes back just like inhibin and negative inhibits the release of luteinizing hormone in the pituitary, and it also negative inhibits the release of gonadotropin releasing hormone from the hypothalamus. Now, in the case of Klinefelter syndrome, if dysfunctional Sertoli cells are not producing inhibin, so if this is not here, this is not happening, and this is not happening, as a result, follicle stimulating hormone is going to be increased because there's not this negative feedback. So you're going to have actually high levels of follicle stimulating hormone in these patients. Now, because Leydig cells are dysfunctional, they cannot produce as much testosterone. So the level of testosterone is significantly decreased. And as a result of that, you don't get this negative feedback. As a result of not having this negative feedback from testosterone, you're going to have severely high levels of luteinizing hormone. So when you get the labs on these patients, the classic is they're going to have low testosterone, low inhibin, and because they have low testosterone, they'll have high luteinizing hormone. Because they have low inhibin, they'll have high follicle stimulating hormone. And since they have low testosterone, they'll have high estrogen. Now, this combination of low testosterone, high estrogen contributes to a lot of the physical characteristics that these patients present with. Normally, they're normal. They have normal IQ. However, intellectual disability is possible. They have decreased testicular mass. That's part of the syndrome, and then also as a result of decreased testosterone. They have a female or a unicoid body shape, And what this means is that they're tall, slim, and often underweight. So Klinefelter's patients are usually taller, and they also have longer extremities. They're also infertile because of decreased testicular development. The testicles are the site of spermatogenesis, where sperm is produced. And then as a result of this decreased testicular mass and decreased testicular development, they are often infertile. They'll have gynecomastia, which is a result of high estrogen and low testosterone, and so enlarged breast tissue. And then they'll also have a high-pitched voice, which is a result of low testosterone as well. Lastly, we'll talk about Turner syndrome, which is an example of a monosomy. It's actually the only monosomy consistent with life, so any other type is fatal. And it may be due to mosaicism or other errors. There's a number of different causes for Turner syndrome. That's not as high yield. What's more high yield is these facts we're going to go through here and on the next slide. So since it's a monosomy, it's 45 chromosomes instead of 46. And the problem is that there's a missing X chromosome. So there's only one X chromosome. So again, there's no need for a plus here because it's not like Downs where there's an extra 21. Symptoms are because of ovarian failure due to ovarian dysgenesis. So briefly, just like we did for Klinefelters, we'll talk about very basic endocrine physiology for the female. So hypothalamus, which also releases gonadotropin releasing hormone, GnRH, which then stimulates the pituitary to then release follicle stimulating hormone and luteinizing hormone, which then go on to stimulate various processes in the ovary, which then result in producing estrogen. We won't get into the other processes because that's beyond the scope of a biochemistry lecture. And estrogen goes on and carries out a number of different functions in the female reproductive cycle and in female development. However, just like testosterone, it comes back and negative inhibits at the pituitary, inhibiting FSH and LH release. Also, negative feedback at the hypothalamus, inhibiting GnRH release as well. The only exception to this is during the mid-cycle of the menstrual cycle. However, otherwise, estrogen comes back and negative inhibits. Now, since you have decreased formation of the ovaries and ovarian failure, you have severely decreased estrogen. You lose this negative feedback, and as a result of that, the pituitary secretes FSH and LH uninhibited, so you have increased levels of FSH and LH. The other thing is, since you have decreased estrogen, this causes primary amenorrhea, leading to symptoms of early menopause much earlier in life. And so if you do the labs on these patients, they're going to have low estrogen, and as a result of that, they will have high LH and high FSH. Clinical features, these are all very high yield, and you definitely want to be aware of these when you're reading the stem, and then often you can get asked about them as well. So streak ovaries or hypoplastic ovaries, early menopause, and then short height. These are all the result of decreased estrogen. Aortic coarctation, very high yield for Turner syndrome. Bicuspid aortic valve, you could hear a systolic click on physical exam. Again, this is very high yield, very common in Turner syndrome. Arched palate, broad, like a shield-shaped chest, wide nipples, cystic hygroma, webbed neck. This is very common for Turner syndrome, and it's due to a lymph vasculature defect in the neck, causes this appearance, and then lymphedema. Horseshoe kidney, they can have these where you have a single kidney joined at the bottom instead of two kidneys. So you have a problem with development like this. Two kidneys that are horseshoe like this versus having two separate kidneys. And then they're usually infertile because of the lack of estrogen and lack of ovarian development. And then they're also at an increased risk of gonadal cancer. All right, so that closes out our discussion of the high yield examples of aneuploidies.